Okay, hi everyone. I am Batul Marzouk. I'm a computational biologist. I'm affiliated with the University of Liverpool, the UK. And just to give a little bit of introduction about the ISB Academy for people who are not familiar with the ISB Academy. So ISB Academy is part of the ISB, International Society of Computational Biology. And what we aim to do within the Academy is to provide talks, seminars that address the latest advances in the field of computational biology. We do have different training series. We have a COVID, indigenous voices. We have a webinars in conjunction with the COSI, and we started a tutorial series just last year to provide more hands-on. So housekeeping, we do have a code of conduct, but so if you do experience or witness any unacceptable behavior, have any other concern, please do report it to the code of conduct committee at this email. We also trying to make our materials as fair as possible, like findable, accessible, interoperable, and reusable. So what we do is we register them online. We make them try to make them reusable for trainers. We reduce the DOI to make it more citable. And we have a GitHub repository for all the tutorials within the ISB Academy. So we have this GitHub organization account where it has all the tutorials and people can go back to the previous tutorials and go through them one by one. We also have the Zenodo community, and by Zenodo we mean a general purpose repository that we add the slides to it. So to make a lot of these tutorials have slides or talks. So what we do, we add these talks within the Zenodo to provide a DOI or make them citable. So as you can see, if you go through any of these and click on any of the talks, you'll prompt you how to cite that talk if you want to reuse anything within the tutorial itself. And with that, I'm very, very, very excited having Meghana and Jury with me to really go through the immune cell profiling from single cell RNA with R. I'll hand it over to you, Meghana and Gauri.
Hello, everybody. Yeah, thanks for today's the talk and the invitation from ICB. We are very excited to go. And just a brief introduction about myself. So I am Meghna Kshirsagar. I joined this as a research fellow in the University of Limerick, and I'm being into this position as research fellow and doing lots of investigation currently into the single cell experiments, and it's only just being 6 months. So basically, I am also onboarding, and with that, I'm also getting acquainted with all the libraries and how to set up the first experiment. So in today's session, I, along with my PhD student Gauri, we are going to discuss what could be like a guidelines of how to set up the single cell experiments, how to do it in a way that's and I mean how to do it in a way that can really be uh, that can really make a difference, and like how we use these results and how we can build up from these results. So let's get started. So the title of today's tutorial would be immune cell profiling from single cell RNA with R, and we'll come to know like why we gave this talk as a immune cell profiling. Why is this title because of some reason? So we'll get to know that during our journey into the tutorial. So we'd like to we have used in our tutorial the data set that was available on the 10 genomics platform. We have used Python, we have used CRAT pipeline, we have used some of the libraries from Bioconductor. Everything we have done is in the R Studio, and all the analysis of the experiments that have been written from the single cell experiment, we have analyzed those results using on the Neo4j platform. So we have made lots and lots of tools, incorporate lots and lots of tools into our experiment, and all our results are also available on the GitHub. So like Batul said, it will be available on the ISCB Academy GitHub for reproducibility. So let's get started. So like in a tutorial, when we are discussing certain concepts of the tutorial, there may be some one in the audience, or so someone who may have alternate parameter settings that had already been previously explored, and they may be different slightly to us. So if their parameter settings works better than us, so please feel free to speak up at that time, because after all, this is how we learn from one each other in the community. And hopefully we also hope that whatever we have planned during this journey, it will help the audience to get acquainted with many new libraries and many new concepts. So we are looking forward to this. So now let's to talk about today's agenda, what we'll be doing. So basically it is a two-part agenda. In the part one, we'll be discussing all about setting up and doing so using the single cell RNA sequencing data. So the first part basically of the workflow is focusing on the single cell experiments, and the second part which will be delivered in a interactive fashion by my PhD student Gauri. So she'll be talking about how to analyze patterns with a very own tool which we name as Allele. Okay, so we'll get into Allele sometime during the midway of this thing. And one thing during the tutorial is like we are the parameter settings that we are going with is from this paper that was being published in Cell Reports, because we are using the same data set that they are using. So so some of the parameter settings comes from this published paper. And then this work that we were doing is also been had also been submitted to BioC 2023 conference that's upcoming in Boston in August 2 to 4, and where we have submitted our work that we'll be presenting today, which was titled Comparative Analysis of Annotation Tools for the single cell types, to get the single test cell types basically. So we'll be presenting that in the upcoming BioC 2023 conference. So what we are going to cover in the part one and the part two, why we are doing the exploratory data analysis. So annotation tools are basically used to in a single cell experiment where you get cells, where the number of scales cells that can scale up very high, like it can start from thousands but it can be millions and billions. So assigning the correct, and human body has 3 trillions of cells, and then those cells comes into various categories, like you have immune cells, you have blood cells, you have tumor cells, and you have all kinds of cells. And again into immune cells, you again have various types, like you have the T cells, you have the macrophages, you have NK dendrite. Then further down, if you want to have a granularity, a fine grain granularity, then you have your the immune cells types are also have their own subsets, like you can have the subsets from T cells, you can have subsets from macrophages and immune and then how basically how the genes are looking into the immune cells, what are the biomarkers in the immune cells, how they are mutated, how they are expressed. And most of the treatment of the immunotherapy also take into consideration this factor. And as most of talk we focus on getting the cell labels for the immune cell types, and then trying to extract the biomarkers, and then seeing what patterns are there in the different immune cells and their subsets, as we are basically focusing on immune cells. So the title of the talk was like immune cell profiling. So basically, we aim to answer two questions from this tutorial, like how do we cluster the cells with the different annotation tools. So do the results from one annotation tool differ from the other? In what manner? And why it is important to get the right cells? So we'll investigate on these things by I mean introducing you to three different category of annotation tools, and then taking an example from each of those. And then when we do the differential gene expression analysis, we again compare like how the annotation tools are extracting the biomarkers. So which annotation tools were able to identify the key biomarkers as and how to, and for now the only baseline that we can compare it is from the findings of the studies that's being done in the past. So then we'll compare the annotation tools just on the basis of the findings that have been reported from different studies. Now, why there is so much of excitement of doing a single cell RNA sequencing experiment? So, as we can see over the last decade, in the last 10 years, most of the studies have been focusing on this. There are platforms that are coming up to do this. To have this high throughput data so that you can go more real, understand more into how the profile of a person looks like from the and then that can help you to design drugs and everything. So lots and lots of been studies have been going on in this direction. Lots and lots of platforms are being developed. Lots and lots of technologies have been developed to make sure that the cells that we are having, so we are able to capture as much as possible the right genome to go towards precision medicine. And this could be one step towards precision medicine. And especially in the last 3 years, during the advent of COVID, we have seen that this a particular method just took up the speed. So, like most of the papers were, but I mean, to design the drug, and the all the drugs that came to the market where there were lots and lots of studies of what's happening in the cells, and whether they'll be able, what kind of drugs needs to be designed to tackle this virus. And so we could see that the studies of the that were reported, it started from like just with the thousands of cells, and they scaled up to millions and billions before they could come to to commercialized solutions. And then it was a really very these kind of studies really made it easier to generate really impactful insights. And then the data set that we are going to use in our experiment today has come from the 10x genomics chromium. So to get more insights into this, we have added a link in the bottom. So it will talk more about like what the, if users want to understand more about this droplet based sequencing, what it is, and how these things was the process, like the flow that's being done, then they can refer to the links. Because biology experts, so we are not discussing more about the biology, but we'll be doing this tutorial from a computational perspective. So basically what we are doing is the file that is being returned, the data that's being returned, it's in the is a matrix of a UMI count. UMI is a unique molecular identifier, and all the processing or all the libraries that we are doing is manipulating this UMI counts for our analysis. So we'll get into that, like how we manipulate this particular matrix. And then how single cell and how this single cell experiment helps us. It has many key applications. It will be able to reveal novel and rare cell types. It will be help to plan targeted therapy. uh, it will be able to discover new biomarkers for for all the cancers. It will be able to profile both healthy and the diseased tissue and the organs. And all of this, so it has applications in very the broader range of applications, be it a pharmaceutical industry or from a research perspective. So let's move to the thing that's of interest for us today, and that's about the UMI file. So UMI, like we said, is a unique molecular identifier, and how does the experimental file looks like initially. So it's basically a raw count of UMIs where we have cells in the columns and we have genes in the rows. So in our particular experiment we had 50,000 cells and we had around 30,000 genes, and that's how we started with our experiment. So basically it's just count matrix, and the count that is there, it is basically the UMI count for that particular gene, which is for the given cell type. So yeah, for any experimental setting, like we see with the COVID-19 example, like we had you could scale up from thousands of cells to the millions of cells. So what is actually the minimum threshold to set up a single cell experiment? Like how many cells do we really need to have? Because most of the cells that could be captured, some of them would be like multiplets, some of them would be like doublets, some would be dead cells. So even after removing that, the viable cells that are really good for the experimentation, how much we need to have. So it again depends on the kind of hypothesis that you will be testing. We can have a less number of cells if you are doing a small hypothesis, I mean just a preliminary just to get familiarized. But for the drug and biomarker discovery, you will be needing, I mean, a large number of good cells, viable cells. So if we look at this, so for the standard skills that's being defined in the studies are like for a single cell gene expression, if we are just your hypothesis is around your gene expression, then anything between 1000 to 10,000 cells are enough. But if your hypothesis is around a single cell gene expression with a high throughput, then you if your platform was high throughput, then you need to have large number of scales for the cells. So you need to have at least 20,000 cells. And because our experiment was single cell gene expression, and the cells that we captured was with the high throughput machine, so we have this more than 20,000 cells. Then the next thing would be like so how many samples you need to have in your study, and this is the most difficult part, as because of the data sharing and all the regulatory rights, it's difficult to obtain samples. But even for to begin with any analysis or any studies, you want to make sure that you have the right samples before you reach any inference or any conclusions. So in an experiment, if you are talking of the homogeneous population where the population is same, then you can always think that each cell is always a replicate, because now the population is same. So in that case, a fewer samples can also be the best, but of course usually we want more than one sample. But in an experiment, when we are doing with the heterogeneous samples, and again we are testing now the treatment, so how it is going to work across different populations, then you need to have more variability. So then if you want to have the more variance, then you would want to have the more replicates. So in that case, the standard recommendation is then you should have, from each of these heterogeneous sample, there should be 3 samples that are homogeneous. And then in all of these samples, because they can be captured during different periods, or during different days, or could be captured using different reagents, or they could be captured using by different sequencing technologies. So one of the thing that would be present in all of the samples would be the batch effects. And in our tutorial, we will be focusing importantly on these batch effects and how we should handle these kind of things that happens because of different technologies, because of different reagents, because of different channels, or whatever. So we need to make sure that we have the right algorithms that takes care of these technical biases. So the data set that we are going to use in our experiment is about it consists of 13 samples coming from for five categories of the gastric antral mucosa biopsies, and this data set is can be accessed using the accession code GSC 134520. And basically you have here the samples are belonging to five categories. So NAG is the non-atrophic gastritis, CAG is the chronic atrophic gastritis, IMW is the intestinal metaplasis with wild level, and IMS is the intestinal metaplasia with severe level, and EGC is the early gastric cancer. So we can see that in most of the case, like for the NAG, we have three samples, for the CAG, we have three samples, for the IMW2, we have two, and for samples. So it's a the three samples are enough to do even a study later on between the course. Like what would be for the persons with the NEG, with the CHG, and with the ISM, or how these could be treated, or like would there be any progression to the cancer. So all of so for these kind of hypothesis, you really need to make sure that you have at least sufficient number of the same homogeneous samples. And then again, the other thing that needs to be taken into consideration should be there should be a variance in the age factor, because age is also going to affect the outcome, or even your treatment plan, that the treatment is going to be planned. So age is going to play an important role, the gender is going to play an important role, other parameters like the person's other behavioral parameters, like whether the person is a smoker, the extent to which he consumes alcohol. So all these parameters also needs to be incorporated into the study. So treatments are not just planned basically by running a single cell experiment, but and just on the basis of the mutated genes, but you need to take into consideration the age, the gender, and most importantly you also need to take into consideration the ethnicity. So each and every the thing is going to count for the right experimental setup. So in the experimental design that we have in a tutorial, we have UM account which we discussed. So that would be the input to the system. Then we would do some, we would process it, like we'll do some filtering things to make sure that we have the cells, the viable cells. So we'll do some quality control and filtering. After that, we'll do the visualization and see what labels each of the annotation tools are labeling the cells. So how the cells get labels, and then we'll perform like the differential gene expression analysis. And again, when we perform this, again we'll have that feedback to the annotation tools, and based on that, taking that information again, they can reassign the clusters, cluster labels. And once we have the cluster labels from the annotation tools, then we can build knowledge out of that. So that would be done with the knowledge graph. So what we have in the tutorial is the use of the R Studio, which basically, if you see here, has these four windows. So window, you have one window where we write the scripts. Then you have the environment where whenever we have each of the object, you will see the object that's being there and all the variables that are being used in the experimentation. Then you have another one which will show you, which will if you are stuck, if you have some errors, then you can always have the library for help. And then what's happening in the session, you have the session window. So basically your R Studio comes like in this the template, and you will be able to do many things here in a very smooth manner. So the first thing that we now do is we download and read the data. See how to download and read the data set. So we'll go to that part. Yeah. So what we do is we are first, before downloading and reading the data set, we need to make sure that we have the right libraries loaded for our experimentation. So we are using some libraries from the CIRAT, and we are using, as you can see, we are using some libraries from the R, some libraries from the bioconductor, and even some libraries that are from the as a part of a dev tools. And throughout this tutorial, we are following the CIRAT pipeline. So making sure that and that annotation the tools for the annotation tools that we are be investigating today, we are investigating three annotation tools. They are the single R, SC, MRMA, and SCSA. So because SCSA is a Python tool, so we will separately show how to do experiments using the SCSA, and then we are going to have a comparative analysis between these three tools on the kind of cell labels each of them have returned. And other than that, there will be some libraries for the visualization, and then some libraries to take care of the batch effects. So that's how we are setting up. So we have to make sure that you have each and every library there. Once the libraries are there, then we'll start with loading our data set. So how we load our data set is by downloading it, which is available publicly. So on the, as you can see on the gene expression omnibus, and with this accession number, so like we previously discussed, we have 13 samples of patients which are diagnosed with gastritis, intestinal metaplasia, or early gastric cancer. So what we do is, once we download this data set, then we combine all these samples together into one object, one CIRAT object. So we are not doing here the we are not initially we don't do like the unless and until we get the cell labels, we don't do the sample wise statistics that we do in the later part of the tutorial. Initially we are interested in getting the cell labels, so we combine cells from each of those patients into one object, and that is this object, the gastric data set dot rds, and we export it as an rds object. Then the next thing is, once we combine all these samples together, then we have to make sure that the sum of the sample, that there may be some cells within each of those samples that may not be of that may not be of any use to us. Like they may be the dead cells, or they may be just the cells that are being repeated. So we have to make sure that we have the right viable cells. So then we do the filtering. So in filtering, we will use the following filtering features. So it could be you can have a filter on the features. The features are the genes. Then you could but then then many of the genes in that the thing would also be the mitochondrial and the ribosome. So we don't want to have those, so we'll drop those also. And once and after having these quality filtering parameters set up, then we can proceed with the second part of the data pre-processing, and that's to run these three steps: the normalization, find variable features, and scaling. And these find variable features are the ones that are going to help the annotation tools to give the labels to the clusters. So we'll see how these parameters are being set up. So now, if we look at the quality control and filtering, we like we discussed earlier in our previous slide, that we are following the recommendations from the authors that have published their study in the Cell Reports. So we'll follow those recommendations in this tutorial. So with this merge CIRAT object, what we do over here is, like we said, we first I mean, categorize these patients. So we get to understand, like from what is the heterogeneity within our data set, within our samples. So like patient 1, patient 2, and patient 3 are coming with are categorized in the sample as NAG. But patient 4, patient 5, and patient 6 are categorized as the samples are coming from CAG, and so on. So we first link that patient to those values. once that is being done, then what we'll do is, on that merged sample where we have samples of all of those 13 patients, we'll set this we will find the pattern of the empty and the pattern with the RPL. And then we'll once we extract this per pattern of empty and per pattern of RT, which is just the mitochondrial percentage and the ribosomal percentage. Once we have this percentage from the merge CROT object, then we set up this filtering parameters. So we say here that we those features, like you have to between the range of 400 and 7,000. Then we want to make sure that we discard those features where the mitochondrial and the ribosome percentage is less than 20. And after setting these filtering parameters, we will get a new CRAT object, that is the filtered object. And then we can go along and we can do all our processing on this merged CRAT filtered object. And then the second part of the data pre-processing would be the normalization. So how we do the normalization is like on the we'll perform basically the library size normalization. This is being done to take care of the sequencing depths. Um, then we do the and usually the standard is the library size of 10,000. So we go with that particular standard. We can see here we are normalizing it to the scale factor of 10,000. So you can see the log normalize. Then we do the find variable feature. So on that filtered object, then we are finding the uh, all the features that have good variance amongst them. Um, and then after that we will be scaling. So we are basically we do this kind of pre-processing, and once we do this kind of pre-processing, then you have the data matrix that is ready for your experimentations, that can be fed into the annotation tools to obtain the cell labels, to cluster the cells. So before clustering the cell, the other thing that we need to do is, because we see that though we filter, though we set the filter range between 400 to 7,000, still there we need to see make sure that we have captured all the features. So because the more the number of features we have, you should also need to have more variance in your data set to make sure that your hypothesis is correct. So what we do is to find out we have captured all the variance in the data set, we run the PCA. So the PCA is used to uh, but it will return a number of principal component features that should be incorporated to make sure that all the variance in the data set is captured. And then there would be again with you can have different settings, but we don't know like we would what would be the number of principal component that we need to pick up. So you could have you could have PCs that can be 50, and that will explain the variance in your data set, or you can have PCs 20 pieces, and that can also explain the variance in the data set. So if we get confused like what should be the right number of dimension that we need to pick choose, then you can rely on the elbow plots. So what we recommended is to have the elbow plots for the PCA. And as we can see, till the 20 it captures all the variance that is present in the data set. So you don't need to have more number of pieces, because when we increase more per pieces, anyway it's we can see that is converging. So after 20 there's not the remaining pieces, they don't have much mm, uh, information, because mostly the features will have lots of similarities between them. So we'll stick with we'll pick this by using elbow plots. So elbow plots are in a way efficient to allow us to choose the number of pieces that's needed for the experimentation. Then once we have done this PCA analysis, now we visualize our data set, our samples. So remember that we have a CRAT object which contains samples of 13 patients, and each of those cells. So like the combined cells that amongst the 13 patients that they have was 50,000 before the but that was made available on the platform. And if we look at the 50,000 cells and how they are clustered, we can see on the left hand side that we have there are some clusters which are predominated by cells coming from one from only one or two patients, and then there are other clusters where we can see that there is a distribution of cells coming from all patients, and that's what we want. But that each of the clusters that we get, they should have the cells coming in from all the patients, because that would explain the biological variation, and we want the cells to be labeled based on the biological variations and not on the technical artifacts. So like we can see on the right hand side also, we had into 5 categories. So if we look into each of those cell clusters, we can see that some of the bigger clusters have cells coming in from a specific group, rather than rather than the cells coming in from all of the groups. So it means that the batch effects are there, and we need to take care of this before we to try to assign labels to the clusters. So we can, what we'll do is now we'll see how we remove these batch effects. Yeah. So the first method that we apply for correcting for taking care of these batch effects is the with the harmony algorithm. So in the harmony algorithm will ensure to remove all the technical variations that are in the samples. And once we run this harmony algorithm, because it is compatible with the CRIT library, so what we need to do here is we have to just run the PCA. We have already done that, and then just use this command, run harmony. So this run harmony command is like supported in the, and then again when we group by patients, we can see now whether the cells whether each of the clusters are having cells coming in from all of the samples. So now with harmony we can see, like after running the harmony, we can see the effect, like now each of the cluster is having a variation, so that there are cells coming in from each patient mostly. So each of the cluster at least have most of the samples represented within them. But is harmony the only method to have the for taking care of the batch effects, or can we investigate other algorithms to see how they perform against harmony? So we went ahead and checked and did the batch correction with another algorithm, that is MNN. Again we did the same thing over here. We use the reduction as MNN. First we did the batch correct on the with the MNN, and then we like substituted the reduction here as MNN. And after doing the batch correction with MNN, we can see here that the clusters that we are now obtaining is that most of the clusters, all of the clusters are having the are having a proper representation of all of the samples. So what we mean to say is in each of the cluster you will find cells coming in from all of the samples. So then this is the way we take care of the batch effects. And now we are ready to do now this is the object that can be used for that is perfect, and has been taken care of the technical variances, or the variances in the data sets, any artifacts, and those have been removed. So we can proceed with this object for performing our differential expression analysis. So we can here we just compare like how it looked like with the MNN and with the harmony. So basically both are good, but we saw that harmony performed much better than I mean MNN performed slightly better than harmony. So we proceeded with this batch corrected object that we got from using the running the MNN algorithm. So now cells, and now we want to give the labels to these cells. So here comes the role of annotations. And now these basically there are two ways that we can annotate these cells. One is through the manual way, and another is through the automatic, and we are restricting our tutorial to the automatic the thing. And how can we reach as near to manual as possible, because the manual way of annotating is the difficult, and it largely relies on the knowledge of the domain expert. So we would not want to rely on that, but we would want to have some tools that can take care of assigning the correct labels to the cells. So we categorize our annotation, but the annotation tools that are available on the by the way they annotate, they are categorized into three different categories. We have the marker gene database tools, we have the correlation based, and we have the supervised classification based. So when we talk of the marker gene database based, so the annotation tools are taking care of some external reference the database in order to give labels to the cell types. So their reliance is on a large number of external data sources that are there. Then the correlation based is like a kind of a feedback thing. And then in the supervised classification based, you have something like the cell typist, which is more inclined towards the manual thing, and we have not used that in our experimentation. So we'll just go with the SCMRMA, single R, and SCSA. So when we do the annotations, now annotation is basically, again, like we say that we had obtained this after batch correction, we had found these clusters of cells, and now we want to give some labels to each of those clusters. So annotations help us to do give the labels. So when we did the annotation with single R, we could the number of cluster labels that they return is what shown over here, of which we were interested in the immune cell types. So we have B cell, CD4, T, CD8, macrophages, monocytes, which we are interested in. So these are some categories of immune cells that were being written with a single R. If we are to see with the cell labels that are being returned with SCMRMA, and SCMRMA makes use of the Panglau DB for the global assignment of cell labels on to the for the global clustering. And if we want to, and if we are interested further to find subsets of any of the global cells, then it has another database. So basically, as we were interested in T cells, what we wanted was also to find the subsets of the T cells. So here the SCMRMA is making use of the Bangalore DB and the T cell AI to label the global cells and then their subsets. And then and then what we want to do is we want to see like when we have the cell labels, coming in from different annotation tools, how many of them are able to identify the key biomarkers that are being reported in the previous studies. So like we said that we are restricting our studies to only immune cells. So we are categorizing the immune cells into T cells, B cells, mast cells, and macrophages, and these are the so some of the marker genes that have been identified in the previous studies. So we just want to see how well they compared against this. So if we look at one of the things is with the single R, when you want to assign some label, and when you are not sure like which the label that has been assigned by any of the tool is in line or not, then what we can do is we can use one more library for visualization, and that's Nebulosa. So what Nebulosa will do, in addition to what's being missing in the UMAP and the TSNE, but that will be taken care by nebulous. If we give it the marker genes that we are interested in, it will and it can combine all the marker genes of interest and see the density of the expression. So where on all of the clusters that's being for found out, it will show the density where it is more predominantly found. And then you can relate this particular image to what was the cell label for this cluster, and that's how we correlate. Like if these were the marker genes that were identified and they were found in that, and they were expressed in that particular cell clusters label that was being given, is this the right cluster for their expression? And then what and once we do this, then we once we have obtained these cell labels, then again we go back to the individual samples, and then we go to samples with the different categories. So for example, you have here a CAG, and then we see what is the profile of the B cells in the CAG. So we can see here what is the profile of the CAG patient for the NK cells, and we can see what's the profile of the CAG patient for the CD8 plus T cells. And then similarly, when we go on building their profiles for each of the immune cells, then later on this kind of diagnosis, when given to the domain experts, they can plan better ahead of depending on their profiles. It could be one of the factor that can contribute to like what kind of therapies can be targeted. So if we go to Yeah, so if we and now see go to this VNet part. So what we did was we explored the annotation tool. So we annotated the cells with the single R. So for annotation, like they also make use of the external database. So single R makes use of this blueprint the data set as a reference data set to give the labels to the cells. So first we have to read in this reference data set, and then you have to convert that object, because now we are running the single cell experiment, so we have to set it up as a single cell experiment. So we are converting now the CROT object that we obtained, the batch cake CROT object, that now we are converting it and setting up to do the setting up as a single cell experiment object over here. Then we go along to annotate the data set with this single R. And once we do this, so ident would be the cell need given. So this set ident will then give the label to these clusters, and then these find all markers would be giving you the it would be returning all the marker genes that are expressed in each of these clusters which satisfy a particular criteria. And we went with the criteria that the log to fold change should be greater than 2, and they should be expressed in a minimum and 50% of the cells. So we found all the markers in for each of those cell types, and we again stored those results as a cluster markers. And further we r- write all the results into a CSV, because that would be very useful for a next part of an experimentation, because it will help us to identify like what were the biomarkers that were written when single R was used to annotate the cells. Similarly is the case with the SCMRMA. So in SCMRMA, like we previously discussed, we have it makes use of the Bangalore DB to annotate and give labels to the clusters. So we can see the the result from the SCMRMA annotation. If we want to obtain the subsets of the T cells, then we make use of the T cell AI reference data set, and it will give you the subsets for the T cells. And then the third method of the annotation, like this is coming from the basically is the Python. So it's basically a scoring based method. So what it does, it is it initially scores, it initially gives some labels to the clusters, and then it scores them. And it may be the case that it may not be able to find some of the clusters, it may not be unsure with some of the clusters. So what it will give in that case would be a question mark. So the question mark is, it doesn't it's not sure whether that particular the label to the cell could be like, if we look over here, it could be like a B cell or a NK cell. So what it does is, then it is basically a feedback kind of mechanism with the SCSA. So it will uh, take after it will annotate the cell labels in two steps. In the first steps, it will annotate, and it will uh, uh, based on the biomarkers. And again in the second case, it will annotate based on the markers that are written from the experimentation. So it will make use of both the reference data set also, as well as biomarkers that are written from our experiment to annotate the cells. And in this way, then we obtain the clusters from the SCSA. And then just to know, like what we miss in UMAPs, are we are not sure whether we just get the cell labels, but we don't know how the biomarkers are expressed in each of those cell labels. So if we want that kind of visualization, then nebulosa would be the right one. So we just show here an example of how the nebuliza can show that what is the density of the biomarkers, and among all the clusters, where is the possibility in which of the clusters the likelihood of them being found or expressed. So we take care of the drawback of UMAPs by using nebuliza. And then later on, once we have these kind of uh, the labels, then you can start building the immune profile. So again, what we did was, we created the data structure table, where we have the cells coming in from each of the patients. So first we check, like for each of the patient, which are the category of cell types that are found. And then we just pick those cells that are categorized into the immune cells or those subsets, and we build profiles. So in this way, we build profiles based on only the immune cells. So you see here the T cells, the B cells, and the NK cells. So basically, why we are using these immune cells only to build the profile is because the major kind of immune cells that are basically reported in all of the studies are B cells, T cells, and the NK cells, other than the dendritic mast cells. And they will play a crucial role in the selection, in how you select which cohort of patient to select for which kind of immunotherapy. So we try to cover in the first part the immune cells, and then their and build the cluster labels. We tried to work only on the cluster labels that were labeled with the immune cells and their subtypes to build in the patient profile. And we saw what was the performance of each of those annotation tools, and we stored all the results of those annotation tools. Now the second part would be from a computational perspective of how we can find patterns and how it could be useful in future, how these patterns can in future play a crucial role in the selection of patients when given a diverse set of immunotherapies available. How can we go towards a more personalized or a precision medicine context? So it will depend on the patterns that are existing amongst the samples, and what could be the likelihood that a particular immunotherapy would have a favorable outcome on that sample. So I'll hand over to Gauri now, who will introduce all about the knowledge base that we have created from our first thing, and that is Ali. So over to you, Gauri.
Hi, thank you very much, Meghna ma'am. So, hi everyone. So, I'm Gauri and I'm a PhD student at University of Limerick. I'm in my second year and I'll be now presenting how we now connect everything from what we have learned from our first part of the tutorial and representation in the form of knowledge graph to extract interesting patterns. So, before we actually start with the interactive demo, let me first take you through the agenda. So, we'll be first understanding why we need knowledge graph in the first place to extract patterns. Instead of, we know there are a we have a lot of different technologies. So, what make knowledge graph different from those? Then we'll see how exactly we can build a graph database with the help of knowledge graphs, and then how exactly the allele looks like and how we can use it to extract patterns. Specifically, we are aiming to answer the question about what are the different entities that are there in the allele, what are their properties, how can we discover them, and how it can be useful to the bioinformaticians, the biologists, and the clinicians. So I have been passionate about knowledge graphs from a very long time. So this is our paper, which me and Meghna ma'am collaborated before starting the PhD. And this was our initial proposal for knowledge graphs. This was not into the exact domain of single cell. It was from a health perspective where we integrated the electronic health records of patients and we extracted patterns. So now we are one step ahead from that, and we are also bringing in the more personalized information of a patient from the genetic perspective. So, what exactly are knowledge graphs? So, just to get started with this, knowledge graph is the collection of the entities and the contextual connectedness between them. So, as you can see on the screen, we can see that this is a very famous example. This is the Wikidata that we see on the internet. So, basically, it is the collection of around 102, 796, and 034 data items. And the beauty of knowledge graphs is that the data can be in more than one format. As you can see here, we also have the textual information of the nodes, but also the images. So knowledge graph beauty of bringing in different types of modalities of data, and that can be connected to each other contextually and also semantically. That makes more sense. And when we see it from a health perspective, so this is a very famous example. This graph is from the COVID-19 bibliography, and what we can see here are the nodes or all the concepts which were related to COVID-19. Now, if we see all the red nodes here, we can see that these are all related to the factual entities, and then the green ones are basically from the SARS COVID-19, the another variant of COVID-19, that is SARS. And as we know, though not directly related, but they were somehow related, and that's where the beauty of knowledge graph comes in. So knowledge graph can find patterns even if two entities are not directly connected. And then if you see at the bottom, the all the blue nodes, you can see that these are mostly the patient specific features, where they are males or they are females, or whether they are adult or middle aged, and so on. So, to summarize, what we can say from the traditional databases, what makes knowledge graph different is the traditional data, when we connect them with each other with some kind of relations, we get graphs. But when we connect a semantic relation with all of these entities, what we get is a knowledge graph. And with knowledge graph, because we have semantics, because we have data, and because we have all these dynamic context, what we can build is a knowledge profile of anything that we want to extract patterns from. So all the studies that we have done in the first part of tutorial, first we build an entire relational database, and then we convert that thing into knowledge graph. So on the top, you can see the literature here. So the literature is basically the previously reported study that we are taking reference from. So the literature for now just gives us the reference of all the biomarkers that we can confirm whether our annotation tools have really found those markers in the from according to the literature. But this literature can bring a lot of things into the knowledge graph on a later stage, because we can go back to the literature to refer back to the all the diagnosis, all the different therapeutic tests, or all the personalized studies that are bringing into. So basically our literature is the source of domain knowledge, or all the source of information that we are aiming for to bring into knowledge graph. The another important entity in our database is the annotation, where annotation represents all the annotation tools that we are aiming for. So for now, we have annotation, we have only 3 different annotation tools, but there exist a lot of different annotation tools which can be further added, and this can also serve as an important entity, important source of information. The cell types are the global cell types that exist in a patient, and the subcell types are the hierarchical division of all the cell types that is connected to the cell types. And finally, we have the genes, and these genes are also the genes are the ones which are the biomarkers for the particular cell types or subcell types. And then moving towards personalized medicine, we also have an important entity called as patient, and that patient would be then connected to all of these knowledge source that we have, and then we can extract patterns of how this patient can be recommended to a therapy, or how he or she can be diagnosed. And so for now we are currently in the testing phase of allele, and this is our entire plan to move towards the proof of concept of allele. So in graph data model, what we have is we have basically nodes and ages. So nodes are the one which represents either the concept, facts, events, and when we connect these two nodes with a age, this is called as relationship, and this relationship can either be directed, or it also can be undirected. So for example, if we see the cell type as T cells as the subcell type, and if we see the global types, we know that T cell is a subtype of immune cell. So if we want to build a relationship from T cells towards immune cell, what we have a directed relationship from T cells towards immune cell, and then it has T cells is a subtype of immune cells. So this is what we mean by the semantic connectedness between two entities. And again, if you want to build a relationship from immune cells towards T cells, so then we can say that immune cells has a subtype called as. So using these basic terminologies, we are going to build the graph database from the relational database that we saw in the last slide. And for that, now building, we need different kinds of knowledge graph tools. Now there exist a lot of different popular knowledge graph tools called as GraphDB, Node2x, TigerGraph, and Neo4j. We are basically interested in Neo4j because of a lot of reasons. First is because it's very easy to plug in into a lot of different programming languages, making it a flexible option from one technologies to another or one programming languages to another. Second thing is Neo4j is also very compatible with servers, which is very useful for scalability. At the same time, there is a beautiful work in the desktop where we can test it out on the local databases storages that we have. So in the initial phase, Neo4j is very helpful in testing the initial proof of concept of Allele. And here are some of the interesting links that you can find for installing Neo4j, because according to different operating systems, there are different files. And as in relational databases, we have the SQL language. Knowledge graph have their own language called a cipher query language. We called it as CQL, which is very easy, as it is very similar to SQL, but it is also very easy to adapt. There are no long queries to write to extract patterns, making it an ideal choice to use Neo4j as one of our tools. So this is actually the Neo4j's local desktop version, how it looks like. So basically on the left hand side, what you can see is all the information that you have in your database. So here we can have different databases. Here you can see all the nodes that are there in your databases, and then you also have the relationships in your database. And then on the right hand side, we can write queries in the Neo4j shell, and then the result of that is seen immediately into the interactive browser. So, so all our information that we saw in the relational model are contained in the type of CSV files, that is the comma separated values files, and those are already we have provided all those files in our GitHub repository, so you can download those. And this needs to be into the import folder of Neo4j, which I'll also show you while we go to the interactive demo. But basically, this is how the cipher queries look like. So basically, we need to import all the CSV files and then build relationship among them, and then a whole graph is built, and then we proceed towards writing queries to extract different patterns from them. So this is an example of how the knowledge graph looks like when we connect all the information that we have from our relational database. We can see that the blue nodes are all the annotation tools. So we have single R, we have SCMRMA, and also we have the SESA. We have the cell types, where these cell types are the global cell types called as immune cells. Then we have the intestinal cells and the stromal cells. And then the subcell type are the subcell types of the global immune cells. For example, for immune cells, if we see, we have macrophages, we have T cells, we have mast cells, and we have B cells. And then the lighter pink nodes are the genes which are expressed in those subcell types. And then we have the literature, that means the study which have previously reported that these are the identifiable genes from those particular subcell types. Now we can see here that though this is a very small small snapshot of what we can bring from knowledge graph, but here itself we directly have 39 nodes. And in real world, this is way more complex and with a lot of different nodes. As we also saw in Wikipedia, the number of nodes is in billions and trillions, and this is very true even in biomedical. For example, this is a genomic KB, which is actually a knowledge graph which is bringing in all the genomic data sets and also the annotations from around 30 different portals. And if we can see here, it has around 318, 790, and 570 nodes. So you can see the complexity that the knowledge graph brings into. But how computational artificial intelligence comes in is because we want to traverse through all these nodes within seconds to bring in extra information that might not be so obvious just by looking all of these different sources of information. So that's where the artificial intelligence comes in. So we will actually see that how we can exactly create nodes, how we can build a relationship, and we'll write a small query to extract patterns from it. So this is our gene file called as gene.csv, and what we have is the header which just specifies the headings of those columns, and what we want now for each of these rows in this file, we want a node to be created. So now what we are going to do is we are going to write this query with load CSV. So this will just load the file and it will create a gene, and it will create a node for each of the gene. And now let us just, and let this will write in the Neo4j desktop now. So this is the desktop version that I just showed now. So here we'll write all the queries, but what I'm going to do is I'll tell you how to automatically create all of these at once. So we also have a ready-made file for it, and this is another node for the literature. Here we'll also do the same thing. We have a literature.csv file, and here also, because we are just referring to for now one literature study, we have the L1 node. So this is how the L1 node looks like. And now once we are done with this, we will actually create a relationship among these two nodes. Now literature describes those genes, that's why we have a semantic relation from literature towards the genes. So for that, what we are going to do is we are going to create. So create words actually creates a relationship, and then we are actually writing the relation and its name. So this R is a unique identifier for the relationship. This can be any variable, and then we write the semantic relationship that we want to have. So here we write describe, and then we have A because that A was our gene here. So this is how the graph looks like once the relations are created. Now what we have done is we have already provided the Cypher query for you. This was the Cypher query, and we have already uploaded to the GitHub. Is we'll just call this file from Neo4j shell, and if we do that directly with all the files, with all the CSV files, if we just click that, it will create all the relations. Now if we go to our this thing again, you can see that now we have everything. We have 39 nodes, and just let us check if everything is created perfectly. Then we are going to check with match n. As we can see that we have all the nodes connected with each other. Yeah. So now we are going to do, okay, there is, okay, let me just do it again. Yeah. So yeah, sorry, it's yeah. So we'll just check this. What we have here. Yeah, we have this in we have this graph. So this is entirely connected with each other. Now what are we going to do is we are just going to extract we are going to extract some patterns out of it. So this is the example that we have to list all common genes written by the annotation tools. What we want is to just check that all of the annotation tools agree upon which type of genes written by them. So this will do it in the actual Cypher query, and let us see how it goes. So what are we is what we need is all the genes which are identified by the annotation tools. So we are going to get all the, we want gene, so we're going to take gene, and because we have a relationship um, with the annotation tool, we are going to get the annotation tools here, and yeah. So when we say G and A, now what we have here is we are getting all of the genes which are returned by all of the annotation tools. But what we want is we only want the common genes which are identified by all of these annotation tools. So for that, what we are going to do is we are going to edit this query a bit. And when we say that we have all the genes and annotation tool, we are going to use these genes And then we are going to get the count of this annotation tools, because we want common annotation tools for each gene. So let's call it as, let's say, count. This with keyword is very important because in Neo4j we cannot write a continuous query. If you want to use any of the variable from our first query, we just have to use the word with, and then we can use that variable further. So let us do that. Then what we want to do is we want to match the annotation tools again, and we want to have the count of, yeah, match the count, and then we will get the count of gene, we'll get the count, and we also have the count of D now as, let's say, ASC. And then we want this G, and we want to have the count of D as ESC. Get this ESC. Is equal to ESC, and we'll return this G and A. Yeah. So here's annotation tool as ASC, and okay, let me just see what exactly G comma A as EC. Yeah, we have this graph now, and now what we are going to do is we are also going to connect this gene types to their original cell types, that where exactly these cell types come from. So we are going to match this match annotation tool, and then we will return the G match. Yeah, now we have this together, and and now we are going to just connect this to their cell types. So here we are also going to use this G is expressed in, because the relationship is expressed in with the subcell type. Yeah. So this in table, we also get the information in the form of a table, and when we click on graph, what we see is in the form of graphs. So this is how all the information looks like in the form of knowledge graph. And yeah, so this is what the testing phase of allele looks like. So in the future, what we are going to do is then to create a proof of concept of allele, with also exploring more annotation tools, because we are more interested into the immune cell types, that brings that, because that can be very helpful in building a patient profile from immunotherapy and therapies perspective. Express our gratitude towards ICB Academy for giving us this opportunity, and specifically to our supervisors, Professor Aideen Kulhain and Connor Ryan, for all the supports and guidance. And we also are very thankful to Bioinformatics and Digital Pathology Group, specifically to our colleagues Maria Doyle, Michael Lynch, Ahmed Al Khan, and Yang Ye, who have a major contribution to our research. So, thank you.
So yeah, thank you. And this is it from both of us, Meghna and Gauri. And hopefully you will be able to follow all our tutorial things. And if you have any, issues, please contact us on our emails, and we'll get back and resolve the issues.
Thank you so much. I mean, this is really, really amazing. Thank you. Thank you for very, very informative, entertaining tutorial. I really can't thank you enough. We're really grateful for the time, for the effort you took to share this expertise with our member. Um, yeah, thank you so much. And for anyone who's joined, please let us know if you got any questions or anything. You can post it as an issue within the repository as well, I believe, or you can get in touch with us. Uh, Thank you. Thank you so much.